So good morning, everyone. I'm Dani Ushizima, and it's my pleasure to be here today to talk about rock science. And what I mean about that is the analysis of geological samples and how we have worked to extend this DOE-funded research to other applications. For example, cell recognition, with the attempts to make advancements in cervical cancer cell recognition. And in order to make this work possible, I've been working for and being a member of 4 main entities. One of them is LBL, a Berkeley laboratory, and I'm actually not in life science. I'm in the computation research division, where I'm a staff scientist as well as a member of CAMERA, which stands for Center for Applied Mathematics for Energy Research Applications, as well as NERSC. I've been with NERSC for the past 7 years. And more recently, I was selected as a BIDS data scientist. Whatever data science means these days, that's what BIDS has been doing, which the Berkeley Institute for Data Science it's at UC Berkeley. So being a part of these entities have allowed me to develop and deploy algorithms to attack a broader community. That's the story I'm going to tell you today. Let me move this. Okay, I think we have a computer freeze here. Oh, great. So a quick overview of what I'm going to talk about first, why HPC has been playing a major role in data analysis, particularly my field with computer vision and image analysis. What does it have to do with rock, what we're doing in terms of analysis of geological samples as well as materials, and how we're bringing that to cell recognition. So first, let me put here on the x-axis. The data is definitely increasing, and we have HPC making possible to develop all sort of different algorithms, particularly here in image analysis, which is a type of data, right? So, and I'm starting digital image processing in 1970. Of course, there is a lot of work before that, but just give you a picture of what's been going on the past 50 years here. In some of the algorithms that are very important in the area that I've been working for, and it doesn't matter if it's geological samples or if it's cells or if it's bones, several of these algorithms can be used in these different areas. I will start with optical flow, and a very famous algorithm is Lucas Conad, which basically you're trying to detect those objects that are moving. It could be a cell, or it could be just your sample evolving in time, and you want to detect which are the variations of these different structures. Multi-scale representation. Well, you may be able to acquire images at very high resolution, but if the structures that you're looking are very large, perhaps you have the opportunity to downsample, so you can represent your image using pyramids, for example. So that can be very helpful. Another type of computation is extraction of texture. One example of computation is to calculate co-accuracy matrices extract a hierarchic descriptors type of algorithm that's very helpful to detect roughness of films, and we do that for actually in collaboration with Intel. We've been working in PMO, which is a type of material to do insulation of transistors. Other algorithms here, Markov random fields, common filters brought to the area of image analysis so that we could model images and deal with the problems like segmentation. Graph cuts is another type of algorithm used in several image problems, in which you model the image as a graph, and very famous algorithms, normalized cuts, proposed by Jitendra Malik, a professor in UC Berkeley. Another type is level set, among the several applications, segmentation. SIFT is a set of descriptors. It stands for a scale invariant feature transform, very relevant for a broad number of applications, and so on. Statistical learning between the 2000 and 2010 really takes off in use of support vector machines and other techniques to deal with the image. So a combination of a machine learning plus what was going on in digital image processing so far, combination of a feature-based classification, and so on. So here, just to illustrate, at the beginning, we have very little data, and you can see that most of the papers will carry Lina as the picture to process. Data is increasing, we can take lots of pictures, and we need more advanced methods, and definitely, we need a lot of HPC to make these algorithms feasible, because some of the users of these algorithms want at least a near real-time feedback. Let me move on. So yeah, the data is very big, and we wanted to analyze geological samples, and we have three main problems here. The volume. All my research on rock analysis, this ranges from gigabyte to terabyte per stack. These stacks are also evolving in time, which means that there's pressure and temperature that need to be measured, while you're capturing 3D volumes, as well as the velocity. We're expecting a 10 terabyte per second as the data acquisition rate. So what we've been doing is to, yes, do a lot of image processing and analysis. We're making the user as part of the loop, but enough to allow that not to be so time consuming, so lots of automation as well. Work on parallelization of these automation so that we can detect our structures. Sometimes these are microcracks, sometimes these are fibers. And when I talk about rock, I'm putting between quotes here. Rock can be geological samples, can be bones, can be composites, materials, and so on. So in order to get my data for the rock science, I've been working with the Advanced Light Source at LBL, and that's the scheme of the accelerator. So each one of these lines are a beam line. So you have an instrument at each one of these beam lines. For the beam line 832, we have an instrument in our Superman here. Superman because he's the guy with the X-ray, so that we can see internal structures of the materials. So what happens is X-ray hits this hutch, the sample is inside this little container, and we'll have access to the structures. This was also, some of the images of that beam line were in Nature Materials in collaboration with UC Berkeley professor Robert Ricci, and he's then postdoc Richard Bale. So together, and also in collaboration with NASA and Air Force, they came up with both the development of the instrument to do the image acquisition as well as the processing of several slides to understand the resistance of a ceramic composite. And this composite, just for if you're curious about, the application is on construction of jet engines, and they need to do fracture analysis. The problem is, at some point, the postdoc left. Unfortunately, we couldn't retain him at LBL. The analysis that was performed mostly manually, that took several months now, was gone with him. So we were approached both by the postdoc and the professor, like, can you do something in terms of automating some of what we've been doing? We have lots of curated data sets. How can we leverage what we learn from that? That's when my team and I came to the project and started working on processing these very high resolutions. What we lacked were both the tools to deal with these data sets, which were very large, and here about 6 gigabytes to 100 of gigabytes, and have the tools. And what's very interesting, most of the users were actually making use of an NIH tool called Fiji. So Fiji is just image A. So here's a way where we're actually talking and leveraging some information from the life science. This project started in 1998 to deal with the microscopy, of life science microscopy, by Wayne Respin, and now it's fully used within at the ALS, the beamline users. So what can we do to improve so that we scale that to the resolutions that micro-CT require? So we accelerate key image processing algorithms here. So these are nonlinear edge preserving filters as well as morphological operators to deal with these high resolution data sets In order to do that, we work on parallel capable algorithms. At this step, we're using Java, OpenCL, and GPU. If you want details what exactly we run in this first stage, we are using an Intel Xeon CPU with the three NVIDIA Teslas, and only with this very humble system, we could achieve 17 times. We could do all our process 17 times faster. Just a little bit of eye candy, and that's, we definitely kind of on NERSC for that, for doing all the visualization, so that's not detecting the structure itself, but providing all the rendering of all these different structures. Here we have the fibers, here we have where the breaks are happening, but we get that for free. No, there's a lot of a processing behind so that we can detect each of these structures individually. And if you want more information, here's a reference. I'll also be happy to talk to you offline about this. And speaking of structure, yeah, there's a lot of math behind detecting these structures, and guess what? This math can be also applied to cells. Not only cells, but different applications, and these are some of the projects I've been working on. Toxin nanoparticles, films for transistors, rocks, and composites. All of them have in common that they are imaged. And at the end of the day, you need to make sense of the content of these pictures. That's where my team and I come to the game. So what about cells? This is a life science section, and we need to move into that really quickly. So the story started with a visiting professor. I had this visiting professor working in my team for a year and a half. Her background was in physics. And I taught her all of the geological samples. When she went back to her institution, to her university, she started talking to pathologists about these rocks and some of the methods that she thought could be applied to cells. I got in touch with these pathologists, and here we are, asking the question, can we use some of these computer vision, computer image analysis to prevent cervical cancer? Can we do a better job in analyze pap smears? So everything starts with a collection of these pap smears, and for all the women here, they will know that you need to go through that at least every three years. Some of them every 6 months. It's not a fun exam, and what we have is that 50% of the abnormal cells can be classified as normal cells. So something's wrong. We're behind on analyzing that. Most of the analysis performed manually, visually. So we do expect that we can bring computer vision to this area and make some improvements. So how that started? We proposed an algorithm last year called superpixel Voronoi diagram. The idea was to screen cervical cells based on pap smears. That was a challenge in a conference, an IEEE conference, which the International Symposium for Biomedical Imaging. And we decided, well, we have some algorithms we've been exercising. What about we get to the competition? Let's get the code tested and compared with others. So that's what we did. We proposed this algorithm, and what we noticed for the database that was provided, which was about 1,000 images, we could analyze each of them using about 12 seconds, and we got the highest precision in comparison with the other algorithms. That fortunately granted us the first place in this code competition. That was the last year, and in 3 days we're going to have the second version. So I'm bringing to you, before I present to that conference in New York, what we're doing. These are some of the results, partial results, and now with a fund by the Berkeley Institute of Data Science. Before, I'm DOE. This is not exactly DOE problem, so I was working on the weekends. Now I get that as part of my weekday job. So just to give you a little flavor, what we're doing behind the scenes, and that's the algorithm, the basis of the algorithm, both of the last year and this year. First, we detect where are the clump of cells as opposed to not cells. So that's what's happening. We're doing some clearing here, using a bunch of filters, cells and no cells, and then we use a basic concept to to individualize these cells, and that takes lower school education. Well, cell is nucleus and cytoplasm. The cytoplasm is always surrounding the nucleus. Bingo. We use that concept to detect these seeds, very high contrast, not much of a problem. We extract a band around this nuclei, and that's the starting point to propagate from so that we can get to the cytoplasm itself. One of the problems of these cells, they overlap. And when things overlap, it's very difficult to individualize. So in the lack of any other information where the borders are, the best way to chop them off is to calculate Voronoi diagrams. And that's basically what we do. That's a great starting point to find where the borders are. If you're laying it into borders that are closer to where they should be, then your computation is much faster, as you look for everywhere in the image, right? So that's exactly what we did as a starting point this year. We do a lot of refinement in terms of finding where the cytoplasms are. Here, let me see if I can show this is not too noisy. So this is a video we put together with some statistics of how complicated it is. It's the 4th cancer among women. That's where these tissues come from, the cervix. These are some of the images we've processed for the challenge last year. So these are real pictures. And who are at danger. So women from 21 to 65 are at danger. As I mentioned, 50% of the abnormal cells will be classified as normal cells. This is an example of the pictures. These are step by step what we're doing. First, we're enhancing the contrast so that we can pick nuclei. Then we identify where are the cell bodies. It's much easier to get rid of the background. There are also other cells that will show up in these exams. The good thing is we do have a prior knowledge of what we should expect in terms of the area of these cytoplasms. We inspect the border, we get some samples of that, and then we can generalize based on those samples where the cytoplasm for each one of these cells should be. This is close to the final result of the last year. Yeah, and we did that for several cells. You're going to have the chance to check a few of them. I promise it's not 961 of them, just to give you an idea how accurate we are. But the way we compute is not by manually looking at all these pictures. There are indexes that are used to measure and compare these different algorithms, and I think that's an advantage of going into competitions and checking these algorithms for particular applications, to have public databases so that we can check and compare algorithms. So our sponsors, and that's it. Well, and with that, I would like to thank our program managers, definitely Lucy Noel and Steve Lee from NASCAR, for the high-performance computing, algorithm development, support, and the work that we've been doing on the geological samples and ceramics and other types of materials. Peter Lee on the BES side for supporting the beamline scientists as well as supporting CAMERA, this new center which combines a lot of efforts from NASCAR and BES. Also for LBL Computation Research Division, the director David Brown and the deputy Jonathan Carter, NERSC Kathy Ellick, which is also an LLD for CS, for whole CS, the director of NERSC Sudeep Duzanji, and BIDS now. Speaking of Nobel Prizes, we have as the director of BIDS, Sol Permuter, and the executive director Kevin Coy. So my big thank you to them so that this cell recognition work could keep going. And with, for those that are not familiar with BITS, here's a little snapshot of who we are. These are the senior fellows. These are the data scientist fellows. We work with a lot of data. We have from people in ecology to people in nuclear engineering, and also social sciences, and all sort of different backgrounds. And we work with a lot of data. If you would like us to take a look at your data, please let us know. We have lots of algorithms. Perhaps we have the solution to your problems. Thank you.